Okay, so our first task is to get you to the point where you understand what action potential is. And, I try to give you some context. So why is action potential important? We are complex creatures living in a changing environment, right? To maintain homeostasis and, frankly, to do anything interesting, we need to be able to react to our environment very quickly. Well, chemistry changes are relatively slow compared to electrical changes, right? Charge can move around a lot more quickly than mass and molecules can move around. So we have evolved this system that takes membrane potential, and potential here is the voltage word. Did you all take physics? Who's never taken a physics class ever? Okay, a few of you. All right, I'll try and make sure that you understand. I'm sort of a closet physicist, but when I talk about potential, I'm talking about electrical difference, okay? So the easiest way to describe that is you've got a battery, right? A good old AA battery that we've used and abused for 100 years now, right? Well, one side of that battery has a bunch of negatives on it, and the other side of the battery has a bunch of positives. That's a potential. Why a potential? Because if we just sit there with the battery in our hand, it doesn't do anything. But if we connect a wire to a light bulb and then back to the battery, something happens, right? So that's potential. We're starting to move current from the negative side to the positive side. All right, so in our bodies, we too have negative side and a positive side, only it's on the two sides of the membrane, okay? And that electrical difference between the two sides can be used to send information or make things happen. For example, make a muscle contract in response to a stimulus, right? Okay. So to build up this idea, okay, first of all, the membrane is selectively permeable to ions. We just covered that. Right? Ions can't pass through the membrane without a channel or transporter of some kind. That means the cell has control over where ions flow and when, okay? Because it put the channels in there. So that's point number one. Point number two, ions carry a charge, so they're affected electrically. This is stuff you learned back in elementary school. Positives repel, opposites attract. Right? So positives and positives push apart, negatives and negatives push apart, positive and negatives are pulled together. Okay, so for example, if all else included, if we had a sodium ion and a potassium ion, the closer we try to bring them, the more they try to push apart, right? Okay, third point, the concentration of ions is different inside the cell than it is outside the cell, all right? Particularly with respect to sodium and potassium, like we talked about just a minute ago, all right? Now, ions diffuse like all other molecules. In other words, from areas of high concentration to areas of low concentration. But this can be affected by their electrical charge. All right, so for example, imagine you had an area that the sodium concentration was very high on one side of the membrane, very low on the other, and sodium could move. Okay, well, obviously, sodium's going to go down its concentration gradient, right? Well, what if on the other side of that membrane there was a whole bunch of potassium? Well, at some point, the sodium would try to go down its diffusion gradient, its concentration gradient, but be repelled by potassium's positivity, by the electrochemical gradient, right? So we can have this kind of balancing act where diffusion forces are countered by electrical forces. So attraction and repulsion can affect diffusion because we have molecules trying to move by their concentration, but are also being affected by the charge that's around. So movement down the concentration gradient can be balanced or stopped by a sufficient electrical gradient or an electrical difference or a charge difference. Okay, so we developed this idea called the Nerst potential. The Nerst potential does not exist in real human cells. Okay, it's a concept. It's a way for you to understand how do cells create a membrane potential in the first place. All right. So sodium's concentration gradient is toward the inside of cells, right? High outside, low inside. Cells are leaky to both sodium and potassium. So both can move down their concentration gradients a little, right? So I think I have a picture to draw on. Yes, I do. Okay. So we have sodium is high outside, sodium is low inside, right? So the concentration gradient is that way. Okay. Well, as more and more sodium, more and more pluses come into the cell down the concentration gradient, the cell inside becomes more and more positive, right? So at some point, no more sodium is going to come in because there's too much positivity in there already. Do you see that? So even though there's a concentration gradient, the sodium stops moving because of the charge difference, right? So sodium diffuses into down its concentration gradient until there's enough positive charge that it stops. So the Nerst potential relates to this. Okay, so yeah, it's not very good at avoiding your hand. Okay, hold on. So the Nerst potential is how much positive is there, how much positive is necessary to stop the diffusion of sodium down its concentration gradient. Okay, did I say it pretty much like that? Yeah. The voltage across the membrane, so the voltage difference, how much positive inside is there at which the sodium ions stop moving because the electrical difference balances the concentration difference. Okay, so it's like two forces. Concentration's trying to push it down, electricity is trying to repel it back out. At some point, those two things are in balance, and we call that the Nerst potential. Okay, it's real in the sense that we can measure it in an experimental system. So if all that existed in the universe was sodium and cells and water, right, the Nerst potential would be about plus 61. In other words, 61 millivolts, more positive inside than outside. So the sign, the plus, tells us that the inside of the cell is positive. The number tells us the extent to which it's positive, right? And we're calling that the Nerst potential. So when the inside of the cell is plus 61, sodium stops entering down its concentration gradient because it's being repelled by the positivity inside the cell. All right? So that's the balance. Now we can do this same experiment for potassium, but it's reversed, right? Because potassium is high inside, low outside. So potassium, if we open the gates, tends to rush out, okay? Well, eventually, as enough potassium leaks out, the inside of the cell becomes more and more negative, right? Because we're moving positives away. So we're left with a deficit of negative inside. So again, if all that existed in the universe was potassium, water, and cell membranes, the Nerst potential for potassium is about minus 94. In other words, across the cell membrane, there's a significant negative difference. Most of the negative charge is on the inside, and it's resisting the flow of potassium out. Okay, so the Nerst potential for potassium is all about repulsion, right? Enough positive charge to keep the sodiums from coming in. The Nerst potential for potassium is all about attraction. Potassium will keep leaving until there's enough negativity inside the cell to start holding on to it again, right? So they move in opposite directions. So we have the Nerst potential for potassium and the Nerst potential for sodium. So to put it in my picture here, right? So concentration, right? Remember that symbol, concentration, right? Concentration of potassium inside versus outside. It's low outside, high inside. So the vector is that way. So that's the concentration gradient. Each plus that leaves a minus behind, right? So when there's enough minus on the inside, this movement is halted. And that comes about at about minus 94. So when the inside, I'm having trouble with the drawing thing. Hold on. There. So when the minus, when there's enough negative charge, that's what the minus says. When we get to 94, the potassium stops leaving because of the electrical forces balancing the concentration gradient forces.